If you give an artificial intelligence, like the toughest bar exam in the country, it passes in seconds.
Right.
It can write a functional Python app before you've even, finished your morning coffee. But if you give that same brilliant AI a basic, messy biology experiment, it completely
It
freezes.
really does.
Welcome to a deep dive into the bleeding edge of artificial intelligence. Our mission today is to answer this massive, somewhat uncomfortable question. Can AI actually do science? Not just memorize it, but, actually do it.
Which is, I mean, it's a crucial distinction. We're at this point where people just assume that because a language model has read every scientific journal in existence,
Mhm.
you just point it at a disease and it'll spit out a cure.
Like a magic 8 ball.
Exactly.
Eh.
But doing the highly subjective, like deeply ambiguous work of a human biologist is a completely different mechanism than just acing a standardized test.
Okay, let's unpack this. We're basically asking if an AI that acts like an encyclopedia can actually walk into a lab, look at a petri dish, and discover something novel, or if if it just knows how to sound incredibly smart at a dinner party.
Right.
So to figure this out, we're looking specifically at the field of bioinformatics. We've gathered this fascinating stack of sources for you. We've got a rigorous academic paper introducing a testing framework called Bixbench.
A really comprehensive
Yeah.
paper.
And an Anthropic blog post detailing their own biomystery bench evaluation. Plus, this really fascinating hands-on YouTube video from a PhD bioinformatician who goes by a genomics PhD, where she basically tests this tech in the trenches.
And we should probably establish right up front why we're focusing on bioinformatics for this.
Sure.
If we're looking for the very first scientific field where fully autonomous AI discovery might actually happen, this is the perfect testing ground. Because bioinformatics sits right at that intersection of biology and data science.
Because they aren't, physically pouring chemicals into test tubes.
Exactly. It relies heavily on analyzing massive data sets, things like DNA and RNA sequencing. So the lab is entirely digital.
Right.
Because it happens on a computer, we don't have to wait for advanced robotics to catch up. We can build a digital sandbox today, drop an AI into it, and just see if it can function as an autonomous researcher.
Okay, so if the digital lab is already built, how are the smartest AIs on the planet actually holding up? The researchers behind our first source, that Bixbench paper, decided to find out.
They really put them to the test.
They did. They took the absolute frontier models, GPT-4o and Claude 3.5 Sonnet, and threw them into the deep end, and the data shows they are basically drowning.
It's a very sobering reality check. Bixbench was designed to mimic the actual daily grind of a bioinformatician. So the researchers created 53 real-world analytical scenarios.
With like nearly 300 open answer questions, right?
Yeah, almost 300. And to make it fair, they put the AI models into a custom agent framework. They gave them a Jupyter notebook, which is a common digital workspace for writing and executing code, and a Docker container.
Wait, let's translate Docker container for a second, just so we don't lose anyone. That's essentially a standardized digital sandbox, right?
Yes, think of a Docker container as an isolated, perfectly organized mini computer inside your main computer.
Okay.
It comes preloaded with all the specific bioinformatics tools and software packages the AI might need. It ensures the AI has a safe, controlled environment to work in without accidentally breaking the host computer system.
Makes sense.
So they handed the AI this fully stocked sandbox, gave it the raw biological data, and basically said, figure it out.
And the model's just face planted. I mean, the highest accuracy any model achieved on the open answer questions was about 17%.
Very low.
But here's the detail that really caught my attention. They also ran a test using multiple choice questions, but they included an option that basically said insufficient information to answer.
Right, an opt-out.
Yeah, an out. And when given that out, the models performed worse than if they had just guessed randomly. They actively opted out of answering the questions entirely. So, I mean, why does a model that knows so much suddenly refuse to engage?
Well, because real science lacks clear optimization metrics. Let's compare it to software engineering. If you ask an AI to write a Python script, there's a very clear objective metric for success.
Does it run?
Exactly. Does the code compile? Does it run without crashing? The AI gets immediate binary feedback. But in biology, you're navigating deep ambiguity.
Yeah.
A real-world bioinformatics task requires long, multi-step analytical trajectories. You have to clean the data, decide what constitutes noise, choose the statistical model, and interpret what is often a highly variable output.
Wait, so these frontier models are essentially freezing up. It sounds like taking a brilliant, strictly theoretical physics student, dropping them into a loud, unorganized restaurant kitchen, and asking them to bake a souffle without a recipe.
That's a great way to look at it. The theory is there, but the environmental ambiguity completely paralyzes them. There's no mathematical formula that just flashes green and says correct.
Wow.
You might filter out a specific gene expression because you think it's a technical error, right? While another scientist might look at the exact same data and argue that gene expression is the key to the whole disease.
So it's subjective.
Highly subjective.
Yeah.
And when the AI gets lost in that subjective ambiguity, its training actually works against it.
How so?
Modern language models are heavily trained via reinforcement learning, not to hallucinate or make things up. So when faced with the messy, subjective choices required in biological data analysis, the AI overcorrects.
Oh, I see.
It assumes it lacks the necessary information and just freezes up, choosing that refusal option.
Hmm.
And to take your souffle analogy a step further, the AI doesn't even know what a kitchen looks like.
Wait, what do you mean?
Well, this brings up another fascinating failure point in Bixbench. The researchers found that the models actually performed better when they were forced not to generate or look at visual plots.
Hold on, I'm stuck on this plot generation thing. We're talking about multimodal AI models here. They can literally see images?
Yes, they can.
I mean, people use them to identify obscure plants from photos on their phones.
Hmm.
How is it possible that an AI fails to read a simple scatter plot or bar chart that it just drew itself? That makes no sense to me.
It feels counterintuitive, right? Because human brains are deeply wired for visual processing. If I hand you a spreadsheet with 10,000 rows of numbers, your brain shuts down.
Instantly.
But if I turn those numbers into a scatter plot with a trend line, you instantly see the relationship. We rely on visual plots to compress data into something our brains can actually handle.
Right.
But an AI is fundamentally different. AI natively processes data as tokens, so strings of text and numbers.
So the raw spreadsheet is actually its native language.
Exactly. For a language model, looking at the raw numerical array is incredibly precise. But when you ask it to render those numbers into a visual plot and then look at the pixels of that plot to draw a conclusion, you're forcing the AI to translate native high-fidelity data into a human-centric format
Oh, wow.
and then interpret the resulting image. It loses precision in that translation. The AI gets confused trying to read the visual representation of data that it already perfectly understood in its raw numerical form.
That is a wild reversal of how we usually think about data, but, it leaves us with an uncomfortable dilemma. If the AI is failing Bixbench so spectacularly because it gets lost in human subjectivity and human visual formats, we have to ask, is the AI fundamentally incapable of doing science, or is the benchmark itself flawed?
That is the pivotal question right there.
Are we testing the AI on its ability to do science, or just its ability to mimic human scientists?
And if the AI freezes up when forced to navigate the subjective, messy way humans do science, what happens if we remove the humans entirely? What if we only feed the AI pure, hard mathematical ground truth?
Which brings us to Anthropic's Biomystery Bench. And here's where it gets really interesting. Anthropic realized that grading an AI on whether it matches human conclusions in biology is often unfair because, well, human conclusions are frequently debated.
Oh, constantly. The classic example is the diabetes drug metformin.
Right.
It's one of the most widely prescribed drugs in the world. We've used it for decades. Yet if you put 5 leading biologists in a room, they will still argue over its primary molecular target and exactly why it works for some patients and not others.
Ah, it's crazy.
So if you ask an AI to reproduce a human scientist's conclusion on a messy data set, you aren't really testing its scientific accuracy. You're grading the AI on whether it made the exact same highly debated subjective choices as that specific human.
So Anthropic decided to completely change the test. They built Biomystery Bench using 99 questions derived entirely from objective, verifiable, ground truth data.
Meaning no subjective interpretations.
Right. We're talking about raw DNA and RNA sequences. The AI is asked a question like, based on this raw RNA-seq data, which specific viral species is infecting this tissue sample?
And the crucial part is that the correct answer isn't based on a human opinion.
Exactly. It's an answer that was objectively verified by a physical, real-world PCR test.
And when you strip away the subjective human bias and focus purely on that objective ground truth, the results flip completely. Anthropic's Claude model, specifically their Mythos preview, performed incredibly well.
Yeah, the numbers were wild.
But the real breakthrough was its performance on what the researchers called human difficult problems.
Right.
These were verifiable biological tasks that a panel of 5 human PhD experts couldn't solve at all. and the Claude model successfully solved up to 30% of them.
Wait. An AI is solving biological mysteries from raw data that 5 PhDs couldn't crack. I need to understand the mechanics of this. How is it getting to an answer when the human experts hit a brick wall?
It succeeds because it uses strategies that humans simply don't have the capacity to use. The first strategy Anthropic noted was the AI's ability to know when it doesn't know.
Oh, what does that mean in practice?
When the AI was uncertain about a data set, it didn't just pick a statistical model and cross its fingers. It spun up multiple completely different analytical methods in parallel. It ran them all simultaneously and then looked to see where the disparate pipelines converged on a single answer. It mathematically layered different lines of evidence.
I mean, a human could theoretically do that, right?
Theoretically, yes,
Yeah.
but practically, a human bioinformatician would have to spend weeks writing, debugging, and running the code for all those different pipelines just to check a hunch. The AI does it in minutes.
Wow.
But the second strategy is where we see a true paradigm shift. It comes down to how human experts annotate data versus how the AI recognizes patterns.
Walk me through that. What does an annotation actually look like in this context?
So if a human scientist looks at a genetic sequence, they can't just read it natively. They take that sequence and run through an established database to see what other humans have previously labeled it.
Okay.
They rely on manual annotations, basically little digital sticky notes left by past researchers saying, this sequence usually means X. The AI, on the other hand, was using direct intuition to spot raw, unannotated patterns in the DNA sequences itself.
Wait, if the AI is spotting raw sequence patterns that human experts miss, is it looking at biology like it's the raw code of the matrix? When you say it's spotting patterns, is it reading the DNA like the A, C, T, G nucleotides like words in a sentence?
That is exactly what it's doing. We have to remember how large language models function. They are trained on tokens, predicting the next piece of syntax based on massive context windows. To an LLM, the English language is a system of syntax, but so is a genome.
It treats it like a language.
Yes, it treats the sequence of nucleotides as a highly complex grammar.
Oh, it's like a master chess player. A grandmaster doesn't sit there and manually calculate every single possible move on the board one by one.
Right.
They just look at the board and instantly recognize the shape of the position. They see the tension and the weakness inherently just based on tens of thousands of past games.
That is a much better way to think about it. The AI is leveraging vast pre-training. This model has consumed hundreds of thousands of scientific papers. It can connect the dots across structural biology, metabolomics, and molecular profiles all at once.
It's just too much for a person.
Exactly.
Mm.
A human brain simply cannot hold that much cross-disciplinary data in its working memory. The AI is looking at the raw syntax of the biology and recognizing the shape of the answer without needing a human to label it first.
Okay, so we have this fascinating dichotomy here. We've seen the AI fail miserably when forced to mimic subjective human workflows in Bixbench, but we've seen it surpass human experts when allowed to find objective ground truth in Biomystery Bench. But those are both still benchmarks. They're controlled digital rooms. What does this actually mean for a scientist going to work tomorrow morning? What happens when a human and an AI pair up in the real world?
For that, we turn to our third source, which really bridges the gap from theory to daily practice. A PhD bioinformatician who runs the YouTube channel Genomics PhD decided to test Claude code locally on her own machine.
In the trenches.
Right. She integrated the AI directly inside her code editor, VS Code, and asked it to handle a synthetic RNA-seq differential expression project entirely from scratch.
Wait, pause. Synthetic RNA-seq differential expression project. Let's translate that for me and everyone listening. What is the actual task she is asking it to do?
Fair point. Yeah. In simple terms, she's asking the AI to look at RNA sequencing data to compare two different groups of cells. For example, a sample of healthy cells and a sample of sick cells.
Okay.
The goal is to figure out which specific genes are turned on or turned off in the sick cells compared to the healthy ones. That difference is the differential expression. It's a fundamental everyday task in biological research.
Got it. And what I found so compelling about her video was that the AI wasn't just a chatbot in a browser window giving her copy-paste code snippets.
No, got it all.
Because it was integrated into her workspace, it could see her files and execute commands. And it didn't just blindly start writing code, it structured the entire project like a seasoned professional.
It was incredibly organized.
Yeah, it automatically created a clean folder layout, separating the raw data, the analytical scripts, and the final results. It wrote a highly detailed re-adding e-file explaining the experimental design, and it generated a step-by-step markdown checklist for the analysis.
Which addresses a massive problem in modern science, which is reproducibility.
Right.
Human scientists are notoriously bad at consistently documenting their folder structures and project management. If an AI can standardize that process seamlessly, it removes a huge layer of human error from the scientific record. But what really stood out in her test was the AI's adaptability when things went wrong.
Yes. I loved this part. At one point, the AI wrote a script that relied on a package called Tidyverse.
Right. Tidyverse is a modern, highly popular toolkit in the R programming language. It just makes data manipulation much easier.
But the human scientist didn't have that specific package installed on her local machine.
And instead of the AI throwing an error or demanding that she stop what she's doing to install the package, it simply adapted.
It just pivoted.
It dynamically rewrote the entire analytical script to use base R, which is the raw foundational code language that's built into every system, so it would run perfectly on her machine exactly as it was.
It's like a mechanic asking for a pneumatic power drill, realizing the shop doesn't have one, and flawlessly rebuilding the engine using just a basic hand wrench instead.
That kind of dynamic problem solving is what makes it a true co-pilot, rather than just a smart search engine. We saw another example of this when she asked it to update all the generated plots to use a colorblind-friendly palette.
Oh, right.
It didn't just give her instructions on how to do it. It went into the scripts, located the visual parameters, made the changes, and regenerated the visuals instantly. Saving hours of tweaking plot aesthetics is a huge quality of life improvement for a researcher.
But the most revealing part of her test was how it ended. She asked the AI to act as reviewer #2.
Yes, the notoriously critical peer reviewer in academic publishing.
Exactly. She asked it to critique the pipeline it had just built.
And the AI didn't hold back at all. It successfully analyzed its own work and explicitly stated that the analysis was only suitable for exploratory learning.
It roasted itself.
Pretty much. It pointed out high-priority scientific gaps, like missing quality control steps for the raw reads, that would absolutely need to be addressed before the pipeline could ever be considered rigorous enough for scientific publication.
So what does this all mean? When we put all these pieces together, it seems clear that the AI isn't the lead scientist yet. It's not going to win a Nobel Prize on its own tomorrow, but it is the ultimate lab manager.
I agree completely.
It takes away the friction of setting up the digital test tubes, organizing the data structures, and grinding through the boilerplate code so the human can focus purely on the why of the experiment.
That is the perfect synthesis. This human-in-the-loop workflow patches the exact weaknesses we saw exposed by Bixbench. Bixbench proved that AI gets paralyzed by the subjective ambiguity of open-ended science.
Right.
But in a co-pilot scenario, the human provides the guardrails. The human provides the biological intuition, the underlying assumptions, and the quality control. The human navigates the ambiguity.
While the AI does the grunt work.
Exactly. The AI executes the technical heavy lifting, writing the code and running the pipelines in an hour, instead of what would normally take a graduate student days or weeks.
Okay, let's summarize the journey we've taken today because it shifts our entire understanding of what AI is capable of. We started with Bixbench, which proved that AI is definitely not ready to be locked in a digital room to cure diseases completely autonomously. It gets overwhelmed by the messy, subjective nature of human scientific trajectories. But then, Biomystery Bench revealed that when you strip away human subjectivity and give the AI pure objective data, its pattern matching abilities are so profound they can surpass human experts, finding answers hidden in the raw syntax of biology.
Finding that shape.
Right. And finally, out in the real world right now, as a co-pilot, it is completely revolutionizing the speed at which humans can test their hypotheses.
Which brings us to why this matters to you, the listener. It doesn't matter if you're working in biotech, managing a software team, or just trying to understand how the economy is shifting. You are witnessing a fundamental change in the bottleneck of discovery.
The bottleneck is moving.
For decades, the hardest part of bioinformatics, and really any data-heavy field, was the execution. It was writing the code, setting up the computational environment, and crunching the numbers. That execution is becoming cheap and instantaneous. The new bottleneck is the ability to ask the right questions.
Exactly. The human's job is no longer to be the calculator. The human's job is to be the philosopher of the data.
But this raises a profound question about the future of discovery itself. We've seen today that AI is already solving biological problems that panels of human experts cannot, and it's doing so by utilizing a kind of pattern recognition that we don't naturally possess. Reading the genome like a language, recognizing the structural shape of a problem without needing human annotations.
Which leaves you with this final, provocative thought to chew on. We started this deep dive wondering if an AI could walk into a lab and hand us a cure. It turns out the answer is likely yes. But if AI is already seeing biological reality in ways our brains are simply not equipped to perceive, will the next great world-changing scientific discovery be something humans can't even comprehend until the AI dumbs it down for us? We might get the cure, but we might not be able to read the explanation.